In this lecture, we're going to address the different steps in which the sodium-potassium ATPase mechanism operates. And there are four conformational states. Conformation referring to the change in the shape of the ATPase protein. And those four are referred to as E1, E1P, E2P, and E2, where P refers to the domain. The domain refers to the very small location sequence of amino acids on the enzyme, which is the ATPase, that binds one ATP molecule. And when ATP is hydrolyzed on this very small sequence of amino acids, the released phosphate group will form a chemical bond with the aspartate amino acid, also referred to as the residue. In the first state, the E1 state, 3 sodium ions are bound along with one ATP molecule. This takes place inside the cell cytoplasm. And the next step is that the enzyme is phosphorylated. What that refers to is that the enzyme will hydrolyze the bound ATP molecule, and the liberated phosphate group will be attached to the aspartate residue, forming phosphoaspartate. And this process is stimulated by the presence of intracellular sodium. And this is the key stimulant for the activity, for increasing the activity of the enzyme. And recall that when sodium enters the cell, either through what are called leak channels at very low, in relatively low amounts, or when ligand-gated or voltage-gated sodium channels open up and results in a relatively large influx of sodium into the cell, will very quickly activate the enzyme to remove sodium. That will move the protein into what's called the E2P state. And when moving into the E2P state, the ion binding as well as ATP hydrolysis will cause a conformational change, and the enzyme will evert to the extracellular side of the cell so that the bound ion onto the enzyme will then face the extracellular environment. And in this state, the connection, the strength of the chemical bond, or the affinity for the enzyme and sodium will decrease, making it much easier to move into the E2 state, where sodium is released into the extracellular medium outside the cell. And with that release, the enzyme becomes dephosphorylated, which is to say that the phosphate group that was bound to the aspartate residue is released. At this point, the binding site for 2 potassium ions outside the cell are available, and the 2 potassium ions will then bind to the protein. The enzyme is unstable in the non-phosphorylated aspartate state. It's in the non-phosphorylated state because of the release of that phosphate. And as such, the enzyme will revert to a more stable structure, and that's by everting back into the cytosol. And in doing so, the affinity for potassium becomes low, and potassium is released into the cytosolic solution. And so we'll go through these diagrams that illustrate the different states of the sodium-potassium ATPase. And so on the left-hand side, here we see the muscle cell membrane, and we see this illustration depicting the sodium-potassium ATPase embedded within the membrane. And this lower illustration represents what's called the catalytic domain. And that's the small region of amino acids that will bind the ATP molecule and generate a chemical reaction that will release the stored chemical energy housed in ATP by breaking off the phosphate group. And so the binding of ATP, as well as 3 sodium ions that had rushed into the cell from sodium channel opening, will move the enzyme into the E1 state, where we see one ATP molecule bound to this catalytic domain and 3 sodium ions bound to the enzyme. Continuing on this slide, here we have still the E1 state represented as we had on slide 16, and now we have the phosphorylated state. And so following ATP hydrolysis, the ADP portion of the molecule is released into the cytoplasm, and the resulting phosphate group that came off of the ATP molecule will form that chemical bond with the aspartate residue on the catalytic domain of the enzyme. And so once again here, we have the E1P state on the left-hand side, which occurs after the hydrolysis of ATP and the phosphorylation of the catalytic domain. Following this hydrolysis, the enzyme will undergo what's called an eversion and will change shape so that the sodium ions bound to the enzyme will have the opportunity to escape into the extracellular medium, and the phosphorylated state will change the affinity between sodium and the ions, and the protein. And so transitioning to the E2 state, sodium is released, and with the release of sodium, the chemical bond holding phosphoaspartate together will be released. And so what we see here is on the right-hand side, here we have the 3 sodium ions released into the extracellular medium, while the bound phosphate group on the catalytic portion of the enzyme is released back into the cytosolic medium. At this point, the release of sodium will allow potassium to bind to the actual protein itself, as you can see illustrated on the left-hand side here in the E2 state. And following this binding, the unstable nature of the protein will then seek a more stable structure by returning back to its original shape, and in doing so will release the potassium ions back into the intracellular medium of the cell. So this ends our part 4 lecture on the sodium-potassium ATPase activity.